my arm to move, we know that is certainly a signal that is traveling down an axon and is causing a release of something that's causing the muscle to contract, right? I guess we're getting, I try to like not do any neuroscience here, you know what I mean? Like I'm trying to minimize it, but here's something. If you have the cell here, and the thing here is not a neuron, but it's a muscle cell, then what will happen is that when this neurotransmitter is released, this muscle cell will pick it up and it will contract, right? And so the neurons are not, the neurons use action potentials. And in fact, the muscle cells use action potentials as well. But a neuron is not contiguous with a muscle cell. The axon comes down, it terminates in a synapse, and the synapse releases something, and then this cell here, if it's a muscle cell, right? Muscle cells will contract. So that's why I can move my hand. Now we know this is true. We know, for example, that it is not simply this, because if we add a drug, let's say, that somehow interferes with this chemical transmission, right? I no longer can close my hand, right? So we can do experiments to demonstrate that absolutely this is a connection here. And again, I will send you videos that discuss this in a much more traditional way. I'm trying to just keep us really grounded in this sort of, just like essentially like a voltage divider based model, right? It all comes down to this. It all comes down to this. All right. Neurons don't exactly look like this. If you've seen pictures of neurons, they look maybe more like, here's that axon. We know that this terminates here in a synapse. This is the soma. Then we see I mean, if you're drawing it yourself, don't worry about the details. It doesn't really, right? Foot fingers or whatever. Right? We see this other thing, right? When you see a classic picture of a cell in your brain or a neuron, it has this kind of thing. There's this thing here that has this big branching structure, right? And this thing is called the dendrite. Now, normally when you take a class that's supposed to teach you about how the brain works, the very first thing they'll do is they'll say the brain is made out of neurons, and the neuron has a dendrite, a soma, and an axon, right? But in this class, I'm teaching everything backward. I'm only getting to dendrite right now. And I will explain to you why we need a dendrite, simply using the kind of analysis that we've done here. Because you could say, okay, so you have an action potential here, right? The action potential gets here. The neurotransmitter is released, right? You generate an action potential here, and then the action potential keeps going, right? And then what would happen? There might be another cell, then another action potential, and then another cell, and another action potential, right? If you stop to think about it though, you think, why did I need these other cells? Right? If let's say it's a muscle cell here, right? If the goal is to contract the muscle cell, just run the line the whole way. Now, is it going to be a long distance? It is going to be a long distance. Good thing, you have action potential boosters. So distance is irrelevant, right? So this seems, as I've drawn it here, useless. Because it's just that you could have just had the line go the whole way, right? And just have here. Why would we need a connection and another connection and another connection? And it's true. If it was so reliable, these additional steps would be useless. But it turns out it doesn't work that way. It turns out what you have is a situation where this cell is coming in here and it's making the connection there. And another cell is coming here and it's making a connection here and there's another cell here. It's coming down, and it's making a connection here. So this cell here is in fact connected to, in this case, 3 cells, right? But in your brain, thousands of cells. And this one here, in turn, does connect to another cell, but if you can imagine, it's just one of 3, or one of 1,000. And this is how you think, right? Because now we have the ability to do some processing. There's no processing here. I mean, just like there's no information, right, that's being sort of gained or lost in this case. It's just being transmitted through. But now I can do something. For example, I can create a, let's call it a coincidence detector. I'm going to call this neuron A, and this neuron B, and this neuron C, and I'll call this one X. And I will say X generates a action potential, if, right, I'm going to be really logical, only if, right, if A, B, and C generate action potential. Right? So now you have a situation where it wasn't what I showed you before, right? Where it's just this one, this one, this one, this one, this one, right? But all the neurons in your brain look like this. And whether they generated action potential or not depends on what is happening here. Why would this one generate an action potential? Well, if this one generates an action potential, well, that depends on what's arriving at it. And this one depends on what are those dependent on? Well, that also depends on what, right? So it, you keep going back and back. Yes.
Can a cell have multiple axons, or can it transmit to multiple other cells?
Yes, the axons can split as well. So I'm keeping it. Yes, there's one. So let's get to that at the end. Yes, the axon can split. They're called axon collaterals. All right. But let's just do, and we'll get to that right next. People have spent their entire careers figuring out how stuff like this works. And people will say things like, whatever, consciousness and feeling love are all about how this all comes together, right? And there's constant waves of reverberating activity going on in our heads, right? Okay, circuits though, forget that, circuits, okay? Sophomore level circuits, how would we model that, okay? This is how you model it. We would take A and we'd model it exactly like we modeled it before, where we'd model it as a, this is the A. I'm going to model it as this, right? This is, I'll call it the A post, right? It's that compartment right there. This is A post. It has a VM. This is a capacitor. This is the ligand gain ion channel. This is some battery. I'll call this E synapse. Just call this G synapse. What happens is when this generates an action potential, it squirts its chemical here. This chemical makes this open up. Okay. Well, this one's going to be similar, right? See it here. So this is now I'm going to call it B post. It has a VM, and I'm going to draw it the same way. Sorry, I know this is blocking, but maybe you know what I'm drawing, right? It's the same thing, right? We have the E synapse, the G synapse, right? And there's this is the capacitor here, right? And we get a voltage here, right? But to be clear, this is the VM here, right? And this is the VM here, right? And you can see I could draw another one here. Now, here's the soma, right? What is the absolute simplest possible way, right? I could connect them, well, with a resistor. And from here to here, also with a resistor. I'm now going to draw you the soma. Here's the soma. This is very familiar, right? Because this is the standard Hodgkin Huxley. If you want, you can call it DNA. Here's our EK, GK, K, GNA, G, and A. Here's C sub M. And this voltage here is the voltage here. Now we're treating the outside everywhere, right? Voltage inside to outside, so I'm just going to call this the ground. I'm going to ground all the outsides because we're treating all the outsides as ground. And now what I'm going to do, right on the same page, right? It's getting messy, but I like it on one page. I'm going to go like this, that, and I'm going to go like this, and like that. And that's how consciousness works. Okay, that's how everything works. You see, this is connected to here, and then this is connected to here, and I'm not going to draw it, but you can imagine a third one, right? Right? Now imagine this. If this goes, right? So we get a squirt, right? And this goes, right? And this gets some sort of voltage, right? It will contribute to the voltage here. If this goes, it will also contribute to the voltage here. How much? Actually, it depends on this resistance entirely. If this resistance was, by the way, 0, it would perfectly track. Because it's not 0, we get a loss. If for this to fire, we need a 30 millivolt change, and this one contributes 10, and this one contributes another 10, and this one contributes another 10, then the only way I can get this to fire is by having all 3 go at once. This is with the additional branch, right? There's an additional branch here going to the C post, is how you build a coincidence detector.